up, people? I grew my Drew fro out this weekend. I think I probably need a haircut, but that's okay. We're on chapter 10, Photosynthesis, Campbell's 7th edition biology. This is part 1. We're going to teach you all you need to know about the sun and plants and how they make glucose. Let's do it. Photosynthesis converts solar energy to chemical energy. Fantastic. It's not just plants, it's other autotrophs as well, including photoautotrophs, algae, various bacteria, and so on. They need carbon dioxide, they need water, and they use the sun's energy as a catalyst to make sugar, glucose. Okay, so this occurs in plants, algae, various protists, which is a eukaryotic cell, eukaryotic organism, and some prokaryotes. You can see all different kinds here. Fantastic. And yes, the stuff in the ocean, that seaweed, is not a plant. It's an algae. Get it straight, people. Heterotrophs, on the other hand, make their food from other organisms they eat. I'm really hungry right now, so I feel like eating a hamburger. Maybe you should too. They're the consumers. So where does photosynthesis happen? Well, specifically in plants, it's the leaves. Here's the structure of a leaf. Okay. Here's the stomata that are typically found on the underside of the leaf, which allow intake of carbon dioxide and removal of oxygen waste. It also happens to allow water to leave, which will be a lab at a later date on transpiration, but we'll talk a little bit later about stomata. Within those leaves are cells, specifically mesophyll cells, and those cells are the dominant photosynthesizers of the plants. Within those cells we have chloroplasts. Chloroplasts, much like we saw with mitochondria, are double membrane. Inside here we have these stacks of thylakoids, which one stack is called a granum, many are called grana, and one thylakoid is that green pancake looking thing that you see there. Okay? On this membrane is where you find chlorophyll, and hence the reason why they look green. Okay? Now the overall equation for photosynthesis is shown there to you on the screen, and it should look very similar to what you saw in respiration, with one little minor modification. If this was respiration, this is photosynthesis. I hope I didn't just blow your mind there. Chloroplasts are involved in redox reactions like we saw in the mitochondria. Here we see where the hydrogens and oxygens are getting moved to and so on as water gets split. Yes, water gets split. Pretty amazing, huh? So water gets oxidized and carbon dioxide gets reduced. Again, the thing that gets reduced is gaining electrons, and the thing that gets oxidized is losing those electrons. And photosynthesis is broken down into two parts. We'll start with the light reactions, and later the second part we'll talk about the Calvin cycle, sometimes called the dark reactions. The light reactions occur in the grana, okay, those stacks of thylakoids specifically on those membranes, and the overall process involves splitting water. It releases oxygen, hydrogen ions, and electrons. The oxygen gets put out through the stomata, it's given off as waste, and the whole process of the light reactions, its purpose is to make ATP and NADPH, although the ATP levels are very low. NADPH, you'll see, looks very similar to NADH, similar molecule, but it has a P for photosynthesis, or for pimpin', but it's really for photosynthesis, not really even. So don't confuse them. NADH for respiration, NADPH for photosynthesis. The Calvin cycle or dark reaction, we don't like to call it the dark reaction anymore. That's kind of an old world thing. Your parents probably called it the dark reaction. Actually, your parents probably had no clue what that was. They don't call it the dark reaction really anymore because that implies that it only occurs at night or in the dark. It doesn't only occur at night or in the dark, although it's the only one capable of occurring at night or in the dark. Calvin cycle can happen whenever. It often happens at night, but not required to. It occurs in the stroma, the equivalent environment to the matrix of the mitochondria. That's the inside there, the inside region of the thylakoids. And the Calvin cycle's purpose is to form the sugar. It takes in carbon dioxides, uses the energy from ATP and the NADPH, and forms sugar. It begins by forming a 3-carbon sugar, and 2 turns of the cycle you'll see will form 2 3-carbon sugars. Together makes a 6-carbon sugar. 3 plus 3 makes 6. The ATP and NADPH, of course, came from the light reactions. Here's the quick overview. Water enters for the light reactions. It gets split, and the electrons are used to excite and move down an electron transport chain, you'll see, just like we saw in the mitochondria. the oxygen is given off. Those electrons are used to form NADPH and create ATP, both of which go to the Calvin cycle to take a bunch of carbon dioxides and combine them together to make sugar. Now, a bit of background on light energy. So it's electromagnetic energy, and we need to convert that into chemical energy. We talk in terms of wavelength because light is a wave. Okay, and wavelength is the distinct distance between the crests. So from one peak to the other peak is the length of the waves. Okay, and this essentially determines the type of energy that we're talking, whether it's UV or ultraviolet or whatever. So here's the spectrum. We're working with visible light here. Okay, UV down here, infrared there, and so on. And the visible spectrum is what we can see, and that's the wavelengths. Those are the wavelengths that drive photosynthesis. Now these pigments that absorb the sun's energy, you are used to seeing most of these pigments as looking green. Okay, when white light from the sun gets split into its individual colors, it forms a rainbow, as you know, and not all colors of the rainbow are used for energy production. Okay, the reason why the chloroplast looks green and the plant looks green is because that is what's reflected off the plant bouncing into your eye. All the other wavelengths, all the other colors of the rainbow are absorbed into the plant and used for energy production. We can test which colors work best in part by using a spectrophotometer, which sends light through liquid medium, through structures, and tests how much or what colors of light, in fact, are passed through, are transmitted through. And you can determine in that case essentially what happens. So here's trying to create an absorption spectrum. Here we have a green light passing through chlorophyll solution, and you'll notice that a very large percentage of very high transmittance gets through. Where if you take blue light and shine it through a solution of chloroplasts or chlorophyll, very little of it gets through. It's a very low transmittance because most of that light is being absorbed by the chlorophyll. So there's some experiments that help us to identify what light works best for plants. And some of our experiments, you'll have an opportunity to do this potentially if you want. There's three sort of famous ones, and we'll talk a little bit about these. But here's three pigments, okay? Chlorophyll A, chlorophyll B, and carotenoids. These are both green, or this is like a bluish green, and carotenoids are the yellowish pigment. And this is a reflection of the wavelength of which pigments absorb which wavelengths, okay? We know this because of an action spectrum that was created specifically for these chlorophyll pigments in a well-known experiment. Shown here by Angelmann. Okay, Angelmann took algae, and on this algae he exposed it to light that he fractured or broke into its component parts with a prism, and so you see the rainbow being shown over these algae. Well, algae, when they're exposed to light, they photosynthesize and they produce oxygen. He took bacteria that are aerobic, they like oxygen, and laid them all along the algae and looked which areas of the bacteria grew the fastest, meaning which bacteria were exposed to the most oxygen. That portion of the algae was photosynthesizing the most. And what he found was this distribution, what they call the spectrum, where the blue and purple absorb the most into the algae or cause the most photosynthesis and oxygen production. And then the yellow, orange, red also, whereas the green and greenish yellow was very little. Okay? So here we have that action spectrum that was referred to for chlorophyll. Excuse me. Here's chlorophyll A, okay, versus chlorophyll B. Very, very similar. You see it has a long hydrocarbon chain that sticks into the membrane of the thylakoids, but this is the portion with the magnesium ion in the middle that is absorbing the sun's energy. There's other pigments as well besides chlorophyll A and B, as we mentioned, but those vary and are in smaller amounts. When these chlorophyll molecules absorb energy, they excite electrons to a higher excited or higher energy state temporarily. And that higher energy state, you then slowly lose the energy like we saw in the electron transport chain of the mitochondrion. Here we have the beginning of an electron transport chain. Excite the electron, let it slowly lose energy, and use that energy to produce ATP. If you actually take chlorophyll all by itself, isolated, and you illuminate it, you excite it, it gives off light and heat all on its own. So this is that chlorophyll that we're talking about here, shown in purple. This is what we call a photosystem. Okay, in the middle of the photosystem is chlorophyll A. That's the dominant pigment. Around the chlorophyll A, you see all other types of pigments: chlorophyll B, xanthophyll, and all kinds of other things. They absorb different wavelengths of light, and they funnel that energy to the center of the reaction, to the reaction center, the reaction core within the photosystem. The chlorophyll A there has electrons that are bound to it. Those electrons came from water. When water was split into hydrogen, electrons, and oxygen, those electrons were donated to the reaction center to be excited. Please understand that light does not split water. Let me say that again. Light, the sun, does not split water. You can imagine what would happen to our world if that were the case. I'll leave that up to your imagination. In fact, there's an enzyme, and it's not entirely understood how water gets split because it's very, very stable. But that breakage gives off these electrons to the reaction center to be excited. Okay? So we call these the light harvesting complexes, and then the electrons get bumped up or excited. Okay? There's two photosystems in the thylakoid membrane, Photosystem I and II. Although the weird thing is Photosystem II comes before Photosystem I, and this deals with the wavelength of light that the reaction center works most with. One's called P680 and one's called P700. The 680 and 700 deals with the wavelength of light that is absorbed by the chlorophyll in its reaction center. Now, mostly what we talk about with photosystem is called non-cyclic electron flow. Very similar to what we saw in the electron transport chain of the mitochondria, we have electrons funneling down this chain, passed from thing to thing. This is non-cyclic electron flow. It produces NADPH and ATP and oxygen. So here we have light entering, being absorbed. Okay, water gets split into its component parts. Electrons donated to here, the P680, this chlorophyll A in photosystem II. Okay, electrons excited to the primary acceptor, then funneled down sequentially. Those electrons used to pump protons across the membrane, again to generate a gradient to make ATP, just like we saw in the electron transport chain. Although in photosynthesis, not nearly the same amount as you get in respiration. Okay, there's our ATP production. We then take the electrons to P700, excite them again with a new wavelength of light. Okay, funnel those electrons down and grab those electrons in another bucket, in this case NADP+, using an enzyme called NADP+ reductase. It helps to reduce NADP+ into NADPH, and the NADPH and the ATP are going to now go over to the Calvin cycle within the stroma. These are produced inside the stroma to power the Calvin cycle. So a nice analogy for this, of course, is using photons to excite the electrons, passing them down this water mill or this mill which produces ATP, and then grabbing the electrons after further excitation into a bucket, capping it with the hydrogen, if you like, and carrying them off to the Calvin cycle. We're going to stop, and part 2 will pick up after the break.